Hi everybody, my name is Thomas Gear. I am enrolled in UNE's cell biology course, and today is December 31st, 2022. So, for my illustration here, I have drawn a prototypical human cell, which is essentially just a generalized cell, which includes all the basic structures and organelles of a eukaryotic human cell. And I've broken it into three sections, 1, 2, and 3, to represent each of my three macromolecules. So the first one is a steroid hormone, testosterone, and the beginning stages of testosterone synthesis, which is the steroid hormone, occur via lipid droplets, which carry the precursor of testosterone, cholesterol, this blue structure here, into mitochondria, drawn here. Inside the mitochondria, the lipids, or cholesterol here, are converted to a molecule called pregnenolone, which I drew here in green. And the pregnenolone molecules are then transported from the mitochondria to the smooth endoplasmic reticulum, drawn here, in which enzymes convert pregnenolone to testosterone, drawn here in orange. And testosterone is carried from the smooth endoplasmic reticulum to the cell membrane via secretory vesicles, drawn here. The testosterone is released from the cell, where it binds to proteins, which help the hydrophobic testosterone steroid hormones travel around the body. Otherwise, it would have trouble doing this, since testosterone is hydrophobic and wouldn't otherwise travel well in the blood. Ultimately, testosterone passively diffuses into target cells, which helps the overall organism regulate characteristics such as body hair, muscle mass, and fat distribution. So the second section here, I'm going to talk about insulin, which is a protein hormone. And in the beginning stages of insulin synthesis occur via ribosomes located within the rough endoplasmic reticulum, drawn here. These ribosomes manufacture a protein called preproinsulin, which I drew here in blue, which is cleaved inside the rough endoplasmic reticulum to form proinsulin, drawn here in green. The proinsulin is then transported to the Golgi apparatus, which I drew here, via transport vesicles that pinch off from the rough endoplasmic reticulum's plasma membrane. And then from the Golgi apparatus, the proinsulin travels to the cell membrane via secretory vesicles, which I drew here. And inside these secretory vesicles, the proinsulin is cleaved again to insulin. And then as the secretory vesicles fuse with the cell membrane, the insulin contents are excreted outside the cell via exocytosis. And ultimately, insulin binds to insulin-binding receptors in other cells, cell membranes, throughout the whole human body for the purposes of regulating metabolism and cell growth in the organism. And for my last section down here, the intracellular protein that I chose is actin. And the formation of intracellular proteins such as actin is different from the formation of testosterone and insulin in that it is less dependent on the endomembrane system for production, modification, and transportation. So regarding actin, individual actin subunits, which I colored here in green, or monomers as they're also called, are formed by free ribosomes in the cell cytoplasm, drawn here. With the help of chaperonin proteins, which I drew here, the actin subunits fold into their native state in the cell's cytoplasm. And then once folded into the correct tertiary structure, the individual actin subunits polymerize to form long polymer filaments, which I drew here. This occurs naturally, resulting from the subunit's polarity. And then ultimately, actin intracellular proteins determine the cell shape. They allow the cell surface to move around. It allows for muscle contraction. It facilitates transportation of organelles, and it provides the cell with overall structural support. And here are my references for this presentation.